What is antigen? Tell me. पढ़ा तो था कुछ. Don't know? No problem. Antigen, and we have the antibody. What is our own? What is our own? Antigen or antibody? Antigen is our own. Antibody is our own. It means it is a foreign particle. It is a foreign particles, or you can say pathogens. The thing is, whatever which is not ours, is called as the antigen. And antigen must produce the what? Immune reaction. It must trigger the antibody. It must trigger the. Otherwise, it will not called as the antigen. Otherwise, it is not called as the antigen. Are you getting my point? So antigen is a foreign particles which is capable. Write it down. लिख लियो. Fine. So whom those written, don't write. Those not written, just write. Antigen is a foreign particles which is capable. Antigen is a foreign particles which is capable to generate the immune reaction. It means whenever antigens comes, our antibody will see it make the fight. And there will be a generation of response called as the immune response. Clear? I am dealing it in a very superficial way. If anyone is interested, you can visit my YouTube channel. Yes, and you will get the detailed description about these things, because we have to go sometimes according to your syllabus. You have the less time to finish your syllabus. So I'm not going in detail, but yeah, just remember, whenever antigen will comes, antibody will react. This is the basis of one biochemical test called as the ELISA test. Write it down. It is also in your syllabus. ELISA test. ELISA test. Pronunciation varies, my friend. Pronunciation varies according to people, according to the state, according to religions. What is happening? The pronunciation varies. Fine? Yeah, क्यों करते हैं? I will tell you. The ELISA, first you write down the full name. Enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. Yes. Assay. 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 Enzyme linked immunosorbent assay. This is the basically test for HIV. Write it down. This is test for the HIV. HIV is a virus which is causing a disease called as the AIDS, acquired immunodeficiency syndrome. So what we are doing is, we are taking the blood. From suspected individuals. We are taking the blood, and that blood we are going to mix with certain antibody, a preformed lab antibody. Fine. If there will be a reaction called as ELISA positive, that person is having the HIV. If there will be a no reaction, it means negative HIV. No. If blood will contain the antigen, if a suspected person blood will have the antigen, it means it must produce the immune response, means immune reaction. So we will artificially providing the antibody in the lab and checking this, is there any reaction is happening or not. If there is reaction is happening, it means it is HIV positive. If there is no, then HIV negative.
तो उसमें एंटीबॉडीज कौन सा?
एंटीबॉडी हम लोग लैब में प्रिपेयर करते हैं, उसको डालते हैं. नो. Yeah. So your question is that antigen. our body do have the antigen. Our body do have the, you know that RBC surface, there is a presence of antigen. How there will be a blood transfusion? Yeah, how you are grouping the blood grouping? On the basis of presence of antigen and antibody. Antibody is present in the plasma and antigen is present on the cell membrane. Fine. So we do have the antigen. We do have the antigen. But that antigen will not elicit the immune reaction. Why? Because as I said, you know that your immune system, your immune cells knows that who is my on and who is my not on. They have the very clear-cut idea, differentiation between the self and non-self. Yes? Yes? So your immune system will not destroy your cells. Yes? But sometimes, like you all are concentrating and that's why he is not concentrating. So sometimes what happened? Your immune system will fail to recognize your own cell. Fail to recognize your own cell and start destroying your own cell. Start destroying your own cell. That will develop the autoimmune disease in you. Autoimmune disease in you. What is autoimmune disease? Autoimmune disease is a disease where your own immune cell start attacking your normal cells. How? Why and how? How means simple, like as they are encountering the other antigen, they are start destroying you. Why? Because they forget to recognize. They forget to recognize. And that can be happen with so many things, the metabolic disturbance, and also when the thymosin is not secreting in the proper way, then who will make your antibodies to recognize self or non-self? No one knows. It's the thymosin which is basically maturing or making specific. Are you getting my point? So if there will be a cancer, carcinoma in the thymus gland, or if there will be a less thymosin secretions, then these things all will basically cause the problem in the antibody. Your antibody will not recognize your own self and they start getting destroying your own cells, and you will develop the autoimmune diseases. There are so many number of autoimmune diseases. Taking the example, rheumatoid arthritis. Rheumatoid arthritis is one of the autoimmune disease. Then you have the multiple sclerosis is the one autoimmune disease, and there are so many. You got the point? Yes? Yes? Fine. So let's conclude today's lecture. What we discussed, basically we discussed about the types of innate immunity, all those 4 barriers we discussed, starting with the physical barrier, physiological barrier, all of you, physical barrier, then cytokinin barrier. Then we discuss about the acquired immunity. We discussed the basic introduction that acquired immunity is basically adaptive immunity. It is work on the basis of immunological cells, and the prime cell which is involved in the acquired immunity is the lymphocyte. The 2 types of lymphocyte, B and T. B lymphocyte is produced from the bone marrow. T lymphocyte is the thymosin, where the maturation's there and everything is going. Okay. Then we discussed about the types, 5 classes of antibody by remembering the mnemonics GAMED. We can list it down easily, the antibody. We discuss about the lymphoid organs called as the primary lymphoid organ and secondary lymphoid organ. What are the primary lymphoid organs? Fast, bone marrow and thymus gland. What bone marrow is doing? Producing the antibody. What the thymosin is doing, thymus gland is doing? Secrete the thymosin, which is basically helping the fine. Then we discuss about the secondary lymphoid organs, what they are doing. What are the 2 types of secondary lymphoid? What they are doing? They are storing the basically formed immune cells. Clear? Then at last we discussed about the antigen-antibody reaction. We discussed about the test commonly employed for HIV testing. And we discussed about the autoimmune disease concept. Clear? Do you have any doubt? Any question? No? Yeah? Acquired immunity types we'll cover in tomorrow's class. There are 2 types. For you just listen, 2 types. One is called as the active acquired immunity, another one is called as the passive. The active is what you developed over a period of time by fighting with the disease, what sebum developed towards the tuberculosis is called as the Active immunity, passive immunity, what you get from the outside, like the vaccines. Clear? Fine.
बैक्टीरिया है
Yes?
वीवो वाला जिसका बैक्टीरिया है
Yes?
वो स्टेशनरी फेज में तो उस पे आएगा
Yes?
वो तो आपसे आप खत्म हो सकता है
No. You have to fight because your body have the plenty of nutrients. That things, no, listen, good question. That things is on the petri dish where we have the limited energy. We provided the one-time nutrient. But for your body, the unlimited nutrient is for bacteria. They will grow forever. As soon as your body get exhausted, all the energy get exhausted, everything will die, including the bacteria. Fine? But if your body will have the continuously the energy, you are continuously getting the food, bacteria will continuously grow if your immune system is not working properly then. Fine? So thank you with having me. Bye. Have a nice day.